Do you enjoy bananas? Well, did you know you're eating your own kind? We share up to 60% of our genetic makeup with this yellow fruit, which makes you wonder, what other possibilities lie beyond our distant relations with these sweet treats? All 6 kingdoms of life are mapped out by blueprints called deoxyribonucleic acid, more commonly known as DNA. We can think of this as a street. Nucleotide bases, adenine, thymine, guanine, or cytosine, form base pairs, purines to pyrimidines, and groups of these pairs forms a gene. Within DNA strands lie thousands of genes, encoding characteristics including your hair color, nose shape, or even the sound of your voice. Wouldn't it just be great if we could take a gene we liked and express it in ourselves? In 1973, scientists Herbert Boyer and Stanley Cohen took this concept, but rather than expressing a different hair color, they expressed frog genes in E. coli bacteria, birthing the beginning of recombinant DNA technology. This type of vector DNA, called plasmids, are found in bacteria. They are used in recombinant DNA technology because they can replicate independently from the single chromosomal DNA. Plasmids also carry an antibiotic resistance in their genes, which protects bacteria that take it up. The recombinant process simply inserts 2 different genetic materials together, creating a hybrid genetic strand. This can simply be imagined as dipping fries in ice cream. To make a recombinant plasmid, we need to trace some victims, alive of course. First, the same restriction enzyme recognizes and binds to the single restriction site in the vector DNA and 2 sites around the gene of interest. Restriction enzymes cleave those regions. The gene of interest is then glued to the vector DNA by ligase enzymes, like a puzzle piece. Next, transformation introduces recombinant plasmids to bacterial cells via heat shock, making the membrane more porous for plasmids to enter. Sadly, not all bacteria take up a plasmid, so they die because they can't grow in the presence of antibiotics. Now, only bacteria containing plasmids are left. But how do we know which plasmids have our gene of interest? By applying blue-white screening, this can trigger a blue pigment in plasmids that don't contain the gene of interest, exposing the wanted bacteria. Since finding that genetic materials can cross between species, recombinant processes may hold the key to finding an immunity to COVID-19. According to BBC, a 2019 study showed a single gene mutation called STING gives bats immunity to coronavirus. If we further investigate a bat's genetics and find an enzyme that cuts the immune gene in vector DNA, a vaccination may be possible. So let's spend less time predicting the future and more time creating it, because this case won't solve itself.